be with you like a forever chemical. I wanna be with you like a forever chemical.
Hi everyone, I'm Craig Butt with SCIEX, and welcome to another episode of Dr. Craig's PFAS Fireside Chats. The first studies on PFAS in wildlife were published just over 20 years ago, and since that time we've learned a lot about their occurrence and to a lesser extent about their health effects. However, most of these studies have still mostly focused on the so-called legacy PFAS, the perfluorinated carboxylates and sulfonic acids such as PFOA and PFAS. However, with approximately 5,000 PFAS compounds in commercial use, it raises the question of what other PFAS are bioaccumulating and what are we potentially missing. Well, my next guest and her co-authors sought to answer those questions by using some creative non-target analysis techniques to study PFAS in beluga whales from the St. Lawrence Estuary in Canada. Please welcome 𝗙𝗵 𝗕, PhD student in the Department of Chemistry at the University of Toronto, to the show. And in fact, I just learned is doing her research across the hall from where I did my own PhD research a few years ago. All right, 𝗙𝗵, welcome to the show.
Thank you for having me. I'm happy to be here.
So you've got a lot in the paper, and I encourage everyone to kind of click on the link below, get into it. But first of all, in terms of your major findings, you detected a large number of legacy and these so-called novel PFAS in the beluga whales. What did you find?
So we decided to take a non-targeted and suspect screening approach to analyze the livers of these beluga whales from the St. Lawrence Estuary. And we found a large array of different legacy PFAS in these whales, which was to be expected. But we also were able to detect many different types of newer PFAS alternatives or novel compounds in the samples. So for example, we found some fluorotelomere carboxylic acid compounds. We found some sulfonamides, many different types of sulfonamide compounds. And also we found some hydrogenated analogs of some of these PFAS compounds that we typically see. And a lot of these compounds that we detected were shorter chain PFAS, which we know are more replacement compounds for the older, longer chain legacy PFAS. So these were pretty interesting compounds to find in the wildlife.
Yeah, absolutely. And so can you give us a sense in terms of the number of different PFAS classes that you found? We know that there's often different chain lengths of these classes, but I mean, how many distinct classes were you able to detect in the beluga whales?
Yeah, so of the 54 PFAS compounds that we detected, there was quite a bit of structural diversity among the different compounds, so we decided to group them into 9 different classes based on their structural features. So, like I said, we detected those legacy PFAS compounds, so PFAS, PFOA, and FOSA, the ones that we typically find in wildlife samples. But just to kind of highlight some of the novel compounds that we detected, firstly, we found those hydrogenated substitutes for those carboxylates and sulfonates and sulfonamide compounds. So those are analogs of those legacy compounds that have a hydrogen atom that's substituted in for one of the fluorine atoms. And the source of those compounds is still pretty unknown. It's kind of up in the air at this point, but the discovery of those in wildlife was pretty interesting. It was unexpected for us to find. We also saw some long-chain fluorotelomere carboxylic acids, as I mentioned. So those are also abbreviated as FTCAs. And we saw those compounds ranging in chain length from 10 to 14 carbons. And with those compounds, we have 4 of the fluorine atoms substituted with hydrogen atoms. And we know that a lot of those compounds, particularly the even chain length FTCAs, have been seen in the environment before. They're known degradation and metabolism products of PFAS precursors. But we weren't really expecting to see the odd chain FTCAs in our samples. So that was pretty interesting. And then lastly, we saw a lot of novel sulfonamide compounds in our samples. So a lot of those compounds that we saw were actually short chain, so carbons ranging from 4 to 7 in chain length. And we saw some of those shorter chain sulfonamides being detected at pretty high detection frequency. For example, the 𝗵𝐵CA, which is the C4 sulfonamide, was detected at about 90% frequency in the samples, which is quite high. And we also saw some sulfonamide-derived acids, some of those short chain as well. So this kind of indicates that there's probably an emission of a broad set of sulfonamides into the environment.
Yeah, no, really interesting because, yeah, as I mentioned, for a long time we were just really interested in two classes, right? Carboxylic acid and sulfonic, and fully fluorinated, right? And you said that you're detecting things that are missing a fluorine, right? And so
That's right. Yeah.
So clearly, you know, your work has shown many more classes that we need to be concerned about, and then also now this shift to shorter chain compounds, which were supposed to be less bioaccumulative, but clearly are building up in your beluga whales. Yeah. So,
Exactly,
can you give us a sense, just in terms of, I guess, like a relevance of these compounds? You know, you detected a lot of them, but what about the concentrations? How do those relate to the legacy carboxylic and sulfonic acids that you saw?
Yeah, so when we're looking at wildlife samples, particularly types of animals that have a really long lifespan in the environment, so for example, these whales live for a number of decades in the environment, we would expect to find legacy compounds taking up kind of the majority of the PFAS profile. So we're expecting to see PFAS, PFOA, and 𝗪𝗎 to kind of be detected at the highest concentrations. And we did see that here. And like I said, that's just because these compounds have been present in the environment for such a long time. They've been bioaccumulating in these whales for decades now. So that was not unexpected. But the novel compounds, some of them that we saw were present at concentrations ranging from like 1 to 40 nanograms per gram wet weight in the liver. And that compared to the total concentration of the PFAS, which was about 100 to 5,500 nanograms per gram wet weight, that number might seem small in comparison to the total concentrations. But in some cases, these novel compounds, with respect to certain classes, did take up quite a large portion of the profile, the concentration percentage profile of the PFAS. So for example, with respect to the sulfonamide class that I mentioned, some of those novel shorter chain sulfonamides actually took up to 20% of the overall sulfonamides profile. So that is a pretty considerable percentage.
All right, so really cool that you measured these compounds and you took this approach. As I mentioned earlier on, very unique to use a non-target approach for wildlife, at least at this point. Now, what do you think about you know, their occurrence in other regions around the globe? Do you think that other researchers monitoring wildlife should incorporate these into their analysis?
Yeah, so like you said, this type of non-targeted approach is pretty new and it hasn't been done that frequently as of yet. But I think that there have been a few recent studies that have demonstrated that some of these compounds are present in other parts of the globe, which I think is interesting to note. So, for example, those hydrogenated PFAS analogs that we saw, especially the hydrogenated carboxylates, there was actually a recent paper that was published after ours that did find those compounds in surface waters in the Netherlands. So that indicates that there might be global emissions in other parts of the world where those compounds are being emitted. And then also with respect to the short chain compounds specifically, we do know that those compounds have been found in the Arctic, especially sometimes at even higher concentrations than legacy compounds.
Right.
So when we're talking about the sulfonamides, specifically the short chain sulfonamides, there have also been recent studies that found FBSA, which is that C4-sulfonamide, in polar bears in the Arctic. So that also indicates that maybe these short chain sulfonamides can also be long-range transported into other parts of the world. And what I find interesting specifically about those sulfonamide compounds is that another recent study from our group did find that when we tested PFAS towards zebrafish, which is a model organism, we found that sulfonamides among all 74 different tested PFAS were actually the most toxic and most lethal compounds towards the zebrafish. So the fact that we are seeing these sulfonamides in these belugas as well as in other animals in other parts of the world, I think is concerning. So I do think that it warrants us kind of paying a little bit closer attention to these sulfonamide compounds specifically.
Yeah. So one of the things I thought was really cool too is that you were able to get you know, a long-term sampling. Right, your samples went back to, I think it was 2000. So you're able to look at some temporal trends. You saw decreases in the legacy compounds, which, you know, I think would probably be expected based on what we know, right? The major sources globally are decreasing. But you also saw some increasing trends, right, for the novel compounds. So tell me about those.
So when we did take a look at the temporal trends, like you said, we found that the legacy compounds were decreasing, which was to be expected. But the short chain PFCAs were increasing across the sampling years, which is probably reflective of the fact that those have kind of ramped up in use since the phase out of longer chain compounds back in the mid-2000s. But then also, some of the novel compounds that we saw, like the sulfonamide compounds, particularly the 𝗕4-sulfonamide, the FBSA, we saw that that compound was increasing a lot over the sampling years. So we actually saw an annual increase of 12% in that compound's concentration across the sampling years. So that's pretty significant and I think concerning, something definitely that should have more attention. And then also the hydrogenated PFCAs that we saw. Although a little bit slower of an increase, we still did see an increase in that those compounds across the years of about 0.5% annual increase during the sampling period. So yeah, we definitely did see some increasing temporal trends for sure in these PFAS.
Okay, yeah. So, you looked at these beluga whales from the St. Lawrence Estuary. What was interesting about this population in the sense of, why did you choose to study this population versus, something else?
Yeah, that's a great question. So these beluga whales, specifically the St. Lawrence Estuary beluga whale population, are really unique from other beluga whales in the world. So they're actually considered endangered by the government of Canada. They have been since 2014. Their population has been declining for decades now to a pretty dangerously low extent, and they're not recovering their population. The reason for us kind of selecting the species is two-pronged. So firstly, we wanted to use non-targeted analysis because we thought that it can help maybe uncover some of the compounds that might be causing their decline and causing their toxicity, because up until this point, we haven't really pinpointed exactly what compounds are actually causing their population decline. And then secondly, since they're so contaminated, we did expect that PFAS would be in their tissue. They have not actually been analyzed before this study for their PFAS concentration in their tissue. So we kind of wanted to use them to kind of biomonitor for the concentrations of PFAS across the years and also further report their levels in their tissue.
Yeah. All right. So we'll get you out of here on one final question, but Holly, you touched on the fact that these are heavily contaminated with a lot of different chemicals. What other chemical classes have been measured in them?
Yeah, so since they have been subject to biomonitoring for a good number of years now, we have detected a lot of contaminants in their species, such as PCBs, PBDEs, DDTs. So lots of different toxic contaminants have been found in their tissue at pretty alarmingly high concentrations. But up until this point, all of those studies were targeted approaches that were used to analyze those contaminants. So we are the first group to use a non-targeted approach to look at these whales. So aside from just looking at PFAS, we're also interested in looking at other concentrations of other contaminants like chlorinated, brominated, and also non-halogenated contaminants. So we're working on that currently, and we're planning on also reporting on the temporal trends of other contaminants in their tissue as well soon.
Cool. All right. So stay tuned. This study looked at non-target approaches for PFAS, but probably a wealth of information out there in terms of non-target for other compounds. So very cool. All right, well, thanks so much. Really appreciate learning about great work being done at the University of Toronto. Obviously, I've got a strong connection there, but really good work, very novel work, and I appreciate it. So,
thank you so much, Craig. I appreciate it.
Thank you.
I wanna be with you like a forever chemical.